Hello everyone, Tanima this end. You're watching this video in my YouTube handle @justtanima_Barbie. Just to let you know, my YouTube venture is totally a personal, anywhere as a controversial social outreach, and it has nothing to do with my profession when engaged with research and teaching in the field of molecular biology and biotechnology in an institution in India. I personally believe that education should be freely accessible to all. And with this in mind, I have initiated a series on cancer biology. This is my 71st video on cancer biology. In continuation to the previous video, the link of which is shared in the description box, the present video intends to further introduce the topic radiation-induced biological effects. The present video partially discusses the paper entitled Radiation-Induced Biological Effects, Molecular and Cellular Mechanisms, and Applications to Radiation, Nuclear Emergency, and Cancer Therapy. The DOI of the same has been pasted in the description box. I would also request you to turn on the subtitle or caption button on the screen. In case you find difficulty with my deliberation, do keep watching. Ionizing radiation includes high-energy photons like gamma rays and X-rays, in addition to subatomic particles such as alpha particles, beta particles, and neutrons. Various forms of radiation demonstrate differences in their penetrating ability and linear energy transfer. Alpha particles have significant ionizing capabilities but limited penetration. Beta particles can penetrate more deeply, yet possess a lower ionizing density, while gamma rays are capable of easily passing through the human body. On our planet, alpha particles can be blocked by paper, while beta particles can be obstructed by aluminium. Nevertheless, in the vacuum of space, the increased energy levels of particles render effective shielding more difficult. The absorbed dose is quantified in Gray or GY. However, the biological effects are more accurately evaluated using the effective dose measured in sievert or SV, which takes into consideration the type of radiation and the weighting factors of the tissues. A brief exposure to around 1 sievert can lead to acute radiation sickness, characterized by symptoms such as hair loss, nausea, and erythema. Exposure to doses ranging from 2 to 10 sievert can lead to considerable patient mortality, whereas doses that surpass 10 sievert are generally lethal unless hematopoietic stem cell transplantation is performed. These results fundamentally stems from the biological processes associated with radiation. Ionization tracks produced by radiation can directly harm DNA, lipids, and proteins, whereas hydroxyl radicals formed through radiation-induced water cleavage facilitate indirect biomolecular damage, which is responsible for about 2/3 of the DNA damage caused by radiation. Unaddressed or improperly addressed damage leads to various cellular outcomes, which are influenced by the dosage and the surrounding environment. These encompass apoptosis, ferroptosis, pyroptosis, necrosis, and various forms of non-apoptotic cell death, in addition to non-lethal consequences like cellular senescence and mitotic catastrophe. So viewers, hope the video could further introduce the topic radiation-induced biological effects. Please don't forget to like and share the video and also subscribe to my channel. Here I would like to mention that the references that were used for preparing the video have been pasted in the description box. And if you're interested to look into the details, you might check the same. Hope to meet you soon with yet another video on cancer biology. Till then, do take care, keep well, and bye.